This video will walk you through the triple sugar iron test. Now, this is a very unique media because it is in a thicker tube and it is a slant, but because of the thickness, it's able to provide an anaerobic environment and an aerobic environment, which is going to be important to the process that we're looking at for this media. So TSI, or triple sugar iron, is going to be a differential media, and it's looking at two things. Can the organism ferment one or more sugars, specifically lactose, sucrose, and glucose? And is the organism able to reduce sulfur and produce H2S? So we do this test specifically when we are trying to further differentiate out the non-fermenter group. And to do this media, we do a stab and streak inoculation. So we use an inoculating needle, get bacteria on the end of that needle, and then we stab the needle into the bottom of the tube, which we refer to as the butt of the tube, and that is the anaerobic environment. And then as the needle is pulled out of the butt region, we then streak the surface of the slant. So you inoculate the anaerobic environment first and then the aerobic environment. And this is important because we want to see, we want to provide both types of environments so that we can ensure that fermentation is going to happen if that's what happens. But if not, they also can do either aerobic or anaerobic respiration so that the organism can survive. Because remember, this is a differential media. We want everything to be able to grow, but we want to be able to differentiate them based on their ability to ferment sugars and then possibly reduce sulfur. So let's take a look at how you make sense of the different possible observations and results. So this media uses phenol red as the pH indicator. And we're pretty familiar with this pH indicator at this point. When the environment is acidic as a result of fermentation, phenol red becomes yellow. If there's no fermentation happening, then the indicator remains red. So we're really looking at the color of the slant and the color of the butt to make sense of what, or to figure out what this organism can do. Can it ferment glucose? Can it ferment glucose and, lactose and or sucrose? Can it not ferment any of the sugars? Okay, so we're looking to see if fermentation has happened. So if fermentation has happened, you are going to see yellow somewhere in this tube, okay? So on the far left, you'll notice that both the slant and the butt are red. So that means that the pH is neutral because fermentation didn't happen. But if you look at the remaining two tubes, you'll see that there is yellow. So we know that at least one of those sugars is fermented. Now in the middle tube, notice that the slant is red and the bottom of the tube is yellow. So you report this as K over A. So K is for alkaline, A is for acidic. The slant is red because it has a neutral or more alkaline pH, hence the K being used to describe the slant. Since the butt of the tube is yellow, it's only yellow because the environment is acidic. So you report that as A. Okay, so the slant is red, so you write that as K. The butt is yellow because it's acidic, so you report that as A. So K over A. What this means is that the organism can only ferment one sugar. Specifically, that's glucose. And that's because glucose is present in the least amount because we're trying to see if the organism can ferment one of the other two sugars. And the vast majority of organisms prefer glucose. If glucose is available, that's what they're going to eat or metabolize. But we want to kind of force them to use one of the other sugars if they can. And so if you look at the middle tube, you'll notice that the butt of the tube is really thick. There's a lot of media in that area. So there's a lot of glucose, lactose, and sucrose. Although again, glucose is present the least. But if you look at this slant region, you see how there's very little media that actually makes up that slant. So because of that low amount of media, that means there's barely any glucose in there at all. So when you stab and streak this media with an organism that can only ferment glucose, okay, it's going to first ferment the glucose. That's always preferred. But once it runs out of glucose in the media, the bacteria's option is to ferment one of the other sugars if possible, or eat anything else in the media that it can to stay alive, right? It doesn't want to starve. So the reason the slant is red is because the organism can only ferment the glucose. So it ate all the glucose in that slant, which originally turned the media acidic. So originally, within about, I don't know, 4 to 8 hours, if you had looked at this tube in the incubator, it would have been completely yellow because it would have been totally yellow while glucose was available in both the slant and the butt. But once that slant was lacking glucose because it had been all used up, the bacteria in that slant region have no choice but to metabolize some other nutrients in the media. But because that process is not fermentation, the pH of the slant increases. It goes back up. So that phenol red converts back to the red color. All right, so that's why your slant is red. It originally was yellow while glucose was being fermented. Once the glucose was gone from that area, the bacteria were eating other things, which increased the pH of the slant, turning it back to red. But because the bottom of the tube has so much more media, there's more glucose down there. And so the bacteria in that area are able to keep eating any remaining glucose. By the time you take the tubes out of the incubator, the bacteria has not consumed all of that glucose. And so that's why the butt of the tube remains yellow because there's just more glucose down there. So the middle tube means that that bacteria is capable of fermenting glucose. Now, if you look at the far right, you'll notice that both the butt and the slant are yellow. So we know that fermentation happened of at least 2 sugars, possibly 3. Now, we can't tell if it's 2 or 3 sugars, but because the slant remained yellow, we know that the organisms were able to consume more than just glucose. They had to be able to consume at least some other sugar that was still present in the slant once the glucose was consumed. So a yellow slant and a yellow butt means that the organism can ferment 2 or 3 sugars. Now you'll also know that there is a gas bubble at the bottom. As a result of fermentation, some organisms also produce gas. And we're able to see that in the media because you'll see how the media came detached from the bottom of the tube due to all the gas that was produced. So with that, you always want to report gas production. And so you would say, the slant is yellow, the butt is yellow. So think about how you would report them. And then you would also write plus gas. because you have to let somebody know that gas was produced. That is unique to certain organisms that can help you identify or differentiate between certain organisms. Those that can ferment 2 or 3 sugars but not produce gas, and those that can also ferment 2 or 3 sugars but also produce gas. All right, let's look at a few other things. So I mentioned previously that we're going to be looking at fermentation, which we just talked about, but we can also look to see if an organism can reduce sulfur as a result of fermentation. So if sulfur is reduced, okay, then this iron indicator that's already in the TSI media is going to produce a black precipitate. And you can very clearly see the black precipitate in the right-hand pictures. And again, that black precipitate happens if H2S is made, because H2S can serve as a final electron acceptor in anaerobic respiration. And so in the media, we have ferrous sulfate, which is the iron indicator that specifically reacts with H2S. So if sulfur is reduced during fermentation, then the ferrous sulfate in the media will react with that H2S and form the black precipitate. So just like with gas, if you see the black precipitate, you need to report it, okay? And so you would report the color of the slant / the color of the butt. So maybe if it's a red slant yellow butt, it would be K/A. And then you would put plus H2S, okay? That's going to be what aligns with your observation of a black precipitate. Now remember, you can only get the gas bubble or the black precipitate if fermentation has occurred. So you never report that you have a black butt. Okay? Your only options are red or yellow, but you would indicate that the black precipitate is present by writing plus H2S. Now keep in mind, Pseudomonas aeruginosa produces piocyanin, which is what makes the blue-green pigment. And so sometimes that blue-green pigment will make the media appear red. But remember, a red slant and a red butt lets you know that no fermentation happens. So you're not seeing H2S, you're just seeing that colored pigment. All right, so then the rest of this is just showing you the different options, which organisms produce which observations and which results. But please keep in mind that with the given organisms that we are working with in this unit, and especially if you're doing this lab in person, your unknowns should not produce a yellow slant and a yellow butt with gas, because when considering the organisms we're working with, you're actually probably dealing with one of the lactose fermenters. And so that would be an incorrect result. Okay, it's a red flag. It means, whoops, you probably misinterpreted something in a previous test. And so you did the wrong one. Because remember, TSI is for our lactose non-fermenters. So you shouldn't get this result, because this is normally indicative of an organism that does produce lactose. So again, here's just the different organisms that can produce these different results. All right, that's the end of this video. Please let me know if you have any questions.